Well, hello, everyone. It's a great pleasure to be here. I'm going to tell you a story about science meets technology meets society today. And this story really starts with our understanding of how information is stored biologically. I think most people here know that that occurs in the molecule called DNA, which encodes all of the information necessary to make cells and organisms. And ever since the discovery of the structure of DNA, which is a beautiful double helix made up of 4 chemical letters that form base pairs, enabling genetic information to be encoded and replicated accurately, scientists have been thinking about how it might be possible to make changes to that code. What if we could not only read the code and write the code and understand the code, but what if we could actually rewrite the code? And that's really the story that I want to tell you about today. It's this question of what if a cell's DNA could be edited just like the text of a document, so that you could actually erase letters, you could erase whole sentences, you could replace sentences, and you could do things that would enable scientists to change the mutations that might cause genetic disease, make changes that allow us to understand the function of DNA in different kinds of organisms, and perhaps enable us to really direct the way that organisms are evolving on the planet. And so the story about CRISPR technology is one of fundamental science that started off as a curiosity-driven research project aimed at understanding how bacteria fight viral infection, how that research project uncovered the tools for gene editing, known as the CRISPR gene editing technology. And that's the adventure that I've been on really over the past several years. And so today what I want to do is I first want to tell you a little bit about the biology of CRISPRs, how they work, what they are, and what they do for bacteria. And then I'm going to tell you about how an understanding of the CRISPR immune system in bacteria allowed scientists to harness it as a powerful technology for gene editing. And then finally, I'm going to turn to a very important issue, that of ethics, how we think about taking a powerful technology that allows us to rewrite the DNA in cells, including our own cells, and how we ensure that that technology is deployed in a responsible way going forward. So let's first talk about the biology. So when a virus infects a cell, it literally lands on the surface of the cell and injects its DNA. And this is a cartoon that shows this occurring in a bacterial cell, where the DNA is held inside the cap of this virus, the top portion of the virus. It's held at very high pressure. And when this virus lands on the cell surface, it injects the DNA very rapidly into the cell, where the DNA encodes all of the information to make new viruses. And that's fundamentally what viruses do, is they take over the cell and they start making more copies of themselves. And for bacteria, this process only takes about 20 minutes. So there's very high selective pressure for bacteria to evolve ways to fight viral infection. And scientists over the last several decades have been uncovering different ways that bacteria do that. But it was only just a little over, maybe about 15 years ago, that scientists first started to understand that there might be a new way, a different way that bacteria fight viral infection that had not at the time been discovered. And that's the system known as CRISPR. So what you're seeing on this slide is a picture of the circular bacterial chromosome, a piece of DNA that encodes all of the information in a bacterium to allow that bacterial cell to grow and divide. And it turns out that in almost half of the bacterial DNA that's been sequenced so far, different kinds of bacteria, they have in their chromosome a sequence known as CRISPR. And these attracted attention even before anybody knew what they were doing, because they had a very interesting property of a series of short DNA repeats, in this cartoon shown as the black diamonds, sequences of letters of DNA that about 40 letters in length that were repeated over and over and spaced by sequences indicated by the colored boxes that turned out to be derived from viruses. And this was the first hint that these CRISPR elements might represent a bacterial adaptive immune system, a way that bacteria can store little pieces of viral DNA in their own chromosome and use that information later to help protect the cell. And one of the hints that this might in fact be some kind of a system like that was the fact that nearby in the genome were located CRISPR-associated, or Cas genes, genes that encoded proteins initially of unknown function, but that always co-occurred with the CRISPR set of sequences. And so scientists had dubbed these CRISPRs, there you can see the acronym there at the bottom, clusters of regularly interspaced short palindromic repeats. And aren't we lucky that it's sort of somewhat of a catchy acronym that we can all remember, at least the acronym, to represent this adaptive immune system. So I want to show you a short video that illustrates how this system operates in bacteria and how it could be harnessed as an immune system. And then I'm going to tell you a little bit more about the biological function of one of the Cas proteins that has been harnessed as the center of this technology. So let's take a look at this video. So we're looking at bacterial cells and viruses arriving in their environment, leading to infection. So viral DNA is injected into the cell, and cells that have a CRISPR sequence have the ability to capture little pieces of viral DNA and integrate them into the CRISPR locus in this very precise pattern that I showed you before, with repeats and viral sequences interspaced. And then the bacterium makes a copy of the CRISPR sequence in the form of RNA, CRISPR RNA. The RNA is chopped up so that each molecule of RNA includes a sequence from a virus and combines with a second RNA called tracer that can bind to the green Cas protein shown here. In the laboratory, the two RNA molecules can be connected to form a single guiding RNA that allows the protein to interact with a DNA sequence matching the RNA molecule. In bacteria, when this occurs, the DNA unwinds and the Cas protein cuts the DNA very precisely at the place indicated by the CRISPR RNA. The broken DNA, corresponding to virus DNA, is then degraded, protecting the cell from further infection. But in plant and animal cells, this kind of DNA breakage is quickly repaired by machinery in the cell that can lead to precise changes at the position of the break. And in this way, the CRISPR system, which started off as an adaptive immune system in bacteria, could be harnessed as a powerful technology for gene editing. So let's now talk a little bit about how this actually works. I like to use the analogy of, I like to call CRISPR a molecular scalpel for DNA. But people often ask me, but what is that? What is it really? I know it's not really a scalpel, so what is it? So let's talk a little bit about what it is. So to understand this, we have to talk a little bit about how genetic information is used in cells. And so as we talked about earlier, DNA molecules encode the information required to make a cell or an organism, and that information is deployed in the form of copies of RNA molecules. These are chemical cousins of DNA that have the same or very similar letter sequence to that found in the DNA. And sometimes those RNA molecules are in turn able to encode proteins. So proteins are made from the RNA, and those proteins carry out all of the functions required to make a cell. But sometimes the DNA encodes RNA molecules that stop right there. They don't encode proteins, but they are functional as RNA. And the great thing, from my perspective, the cool thing about CRISPRs is that these are systems that use both. They use proteins and they use RNA molecules to create a very effective system for finding and cutting up viral DNA. And so the way that this works, so this is a cartoon that summarizes what you saw in the video. So we have a bacterial cell with the cell surface shown by the yellow line, viruses landing on the surface and injecting their DNA. And if the bacterium is lucky enough to have a CRISPR element in its chromosome, it can steal little bits of those viral DNA molecules and integrate them into the CRISPR site in the chromosome. And then a little RNA copy of those sequences is made, and those RNAs are chopped up and they combine with Cas proteins to form a protein RNA complex, an interaction that allows the cell to search for sequences of DNA matching the sequence in the RNA, and if it finds a match, allow the Cas protein to cut the DNA very precisely. So this is probably my most technical slide, but this is really showing you how this system was investigated by my laboratory and the laboratory of Emmanuel Charpentier and our students to understand exactly how it works. So we were puzzled by this. We thought, how does this bacterial cell have a truly programmable protein that can find and cut any viral DNA molecule? And so we studied this and we found out that in many bacteria, there's a protein called Cas9, which in this cartoon is the blue molecule. And this protein is a protein that can be programmed with RNA molecules coming from the CRISPR sequence to find and cut DNA molecules that come from viruses. And the way it works is that the blue protein, the Cas9 protein, grabs onto DNA at places in the sequence that match the yellow sequence in the slide that comes from a virus. And that is embedded, it's part of the CRISPR RNA molecule. And when that occurs, the DNA opens up, and the protein has two chemical cleavers that swing into place and cut the DNA double helix. Now, importantly, when we were studying how this worked in the laboratory, we realized that this protein requires a second RNA molecule, a molecule called tracer, the red molecule in this cartoon. And that RNA molecule is essential for this system to operate because it allows the Cas9 protein to grab onto the CRISPR RNA. And so once we had that understanding, it was possible to combine the two RNA molecules into a single RNA that we called a single guide RNA that had the information to direct this protein to a DNA sequence, as well as the information to recruit it to the Cas9 protein in the first place. And for us, this was really the proverbial moment when fundamental science became a technology, because we realized that this two-component system could be harnessed as a powerful tool to induce gene editing. So to tell you about how this technology works, I first want to show you a little bit more about how the Cas9 protein is truly a programmable enzyme. And this is a 3D printed model that shows the Cas9 protein in white holding on to its orange guiding RNA molecule and interacting with a DNA double helix. And the great thing about this protein and its programmable ability is that it's very easy for a molecular biologist to change the sequence of the guide RNA. It's something that we do routinely in our labs for all sorts of research. And by changing the sequence of the RNA in this system, we can direct this protein to interact with any DNA sequence that we're interested in cutting. And so in this 3D model, what you can see is that the protein and its RNA molecule, its attached RNA, searches through a cell to find a matching DNA sequence. And when that occurs, it opens up the DNA double helix and allows each strand of the DNA to be cut. So it makes a very precise cut at exactly the place dictated by the RNA. And so by manipulating the sequence of the RNA, we can direct this protein to search through the cell and make a cut at any desired place. So you might be thinking, well, that sounds sort of cool, but what does that have to do with gene editing? And this is where we saw a really interesting convergence of an understanding of a bacterial immune system with research going on in a seemingly unrelated field of DNA repair. And so what many scientists had come to appreciate over the past few decades is that when animal and plant cells experience a double-stranded break in their DNA, in one of the chromosomes in the cell, that double-stranded break has to be repaired to avoid damaging the cell or even killing it. And so those cells have machinery to repair the break, leading to either the ends of the broken DNA getting pasted back together, sometimes with a small change in the sequence introduced at that repair site, or sometimes integration of a new piece of DNA at the site of the double-stranded break. And that actually leads to incorporation of new genetic information in the process of repair. And so over the last few decades, scientists had begun to figure out ways to introduce double-stranded breaks into DNA at precise locations that would allow this kind of repair to be harnessed. And so with the CRISPR technology, the ability to program the Cas9 protein with a single guide RNA, something that is easy to do as a molecular biologist, means that we now have a tool to introduce double-stranded breaks at any sequence where we want to effect a change in the DNA of a cell or organism. And we can do that in a fashion that is inexpensive, simple to do, and highly effective. So let me show you, I want to show you another video that illustrates how we imagine that this bacterial system operates once it gets inside of an animal or plant cell. And so we're zooming into a cell, and of course, unlike bacteria, in animals and plants, the DNA in the cell is actually stored inside of the nucleus. So it's kept in sort of a safe place, and furthermore, it's highly packaged. So you can see the blue DNA wrapped up around green proteins, and this bacterial system has to search through all of the DNA packaged in this way to find a sequence that matches the sequence of its guiding RNA. And when that interaction occurs, it opens up the DNA, allows the protein to make a blunt break in the DNA, and then the broken ends of the DNA molecule are handed off to the repair machinery in the cell to effect a change. And in this example, you can see incorporation of a new piece of DNA during the repair process. And remarkably, this system works very well in every type of cell or organism where it's been tested. So it's a very democratizing technology. It really opens up gene editing to laboratories around the world to do both research and various kinds of applications. So let me tell you a little bit about some of the different applications that are already underway with the CRISPR technology. And you have to appreciate that this is a technology that's only about 5 years old. So it's, or not even 5 years old. So it's a very new technology, but it's been deployed incredibly rapidly for exciting applications. One of which is the use in animals and plants that are important for agriculture. And this is actually leading to big questions about how we define genetically modified organisms. And it turns out, as I discovered when I started diving into this, that different countries define GMOs differently. So this is now engendering a lot of debate about how we define these and how we decide to regulate these kinds of organisms that will increasingly be coming to markets around the world. A second example is the use of the CRISPR technology to make changes that will be very important to treat diseases in the clinic. And I think one of the first applications that we're likely to see entering clinical trials, probably within the next couple of years, is the use of CRISPR Cas9 gene editing technology to correct the mutation that causes sickle cell disease, a well-known blood disorder that has a very well-characterized genetic mutation that can now be fixed using this kind of precision gene editing. A third example is the use of the CRISPR technology to create what are known as gene drives. This means being able to drive a trait through a population very quickly. And this is already being deployed in the laboratory for making changes to insects that carry disease. For example, mosquitoes that might carry various kinds of pathogens. And we imagine in the not too distant future the ability to use this approach to create mosquitoes that would be impervious to infection by pathogens and thereby avoid spreading disease in places where it's otherwise very difficult to control these kinds of vectors. Another example is using the CRISPR-Cas technology to understand the fundamental aspects of genetics that control disease in other animals that might be informative for humans. For example, understanding, in this case, how Tasmanian devils are able to fight off face cancer with their underlying genetic traits. A fifth example is using the CRISPR-Cas9 technology to create animals that are going to be better suited as organ donors for humans. Pigs have long been thought of as one of the best animals from which to get organs for donation, but they have various challenges, including being infected by viruses that we don't want to spread to humans, and also having cells that look different when they're organs that look different when they're transplanted into a patient. So that by using gene editing, we can, in principle, create organ donor pigs that will have organs better suited for that kind of application. And then there's the use of gene editing in human embryos, something that has attracted a lot of attention over the last few years, and I'm going to say more about that shortly, and has led to a series of meetings and discussions, including the release very recently of a report from the National Academy of Sciences here in the United States, in which a very distinguished group of scientists reviewed this area and said that, in effect, that human embryo editing should be allowed for research purposes only until the 14-day stage of development, but certainly not for any kind of clinical use, meaning the use of such embryos to create a pregnancy until and unless there is very serious attention paid to the ethical implications of that use and broad societal acceptance. And then there's the ongoing clinical trials that have already started in China and are soon to begin here in the United States to use CRISPR-Cas gene editing to make changes in the immune system of patients that have cancer, to make their immune cells more capable of finding and destroying cancer cells. Sort of the merger of two incredibly exciting areas, gene editing on the one hand and cancer immunotherapy on the other. And then finally, this is a very young technology, so it's still evolving very quickly. And so we find that we're still learning how this technology functions in cells, how it functions efficiently in different kinds of systems, and how we can deploy it in even more effective ways in the future. So finally, that brings me to the topic of the ethical use of gene editing. And I want to in particular focus on the question of what we call germline editing. So many of the kinds of gene editing applications that we currently see being developed quickly for clinical use involve what we scientists call somatic cell editing. So that means being able to make changes to the DNA of an adult in such a way that those changes are not going to be inherited by their children. But there's another kind of editing that we call germline editing. And this really means taking cells that are so-called germ cells, meaning sperm or egg cells, or very early embryos, and making changes to the DNA of those cells in such a way that those changes are transmitted to future generations. And this can be done, we know, in basically we think any type of organism, including humans. In this example here, you're seeing a pipette that's holding on to a fertilized mouse embryo and a needle injecting gene editing molecules on the other side. And we know that this is a very effective way to make changes to the DNA of mice that has allowed scientists worldwide to very quickly use this as a way to create better animal models of human disease. Now, to show you what germline editing kind of looks like, I'm going to show you a very beautiful kind of visual example that was sent to me recently by a student at MD Anderson Cancer Center in Houston, Texas. And so she did an experiment in which she had frog embryos. These are 2-cell embryos. So you're seeing these round 2-cell embryos. And what she did was to inject gene editing molecules into one of the 2 cells in such a way that she could replace a gene important for the brown color of the frog and replace it with a gene encoding a bright green fluorescent protein. And so you see this beautiful image of these 2-cell embryos in which one cell has had this gene editing occur to the DNA and the other cell not. So we get this split right down the middle. And these cells remain, these embryos remain perfectly viable. So they will go on to develop into normal frogs, except that we can see the change in each side of the frog. So on the top image, you can see a tadpole. And if you look at the top half of the animal, it has the normal brown color, but the bottom half is missing the brown pigment. And if we put that animal under a fluorescent light, we can actually see that half of the animal is green. And so this really illustrates the power of this kind of gene editing. Now, this is sort of a cute example, but it has really profound implications, because this means that as human beings, we now have the ability to control our own evolution. We can make changes to our own DNA that will be inherited by future generations. And that's sort of an awesome power. Now, for me, as a research scientist, as a biochemist, somebody who studies molecules and how they work, this really came home to me in early 2014 when a research paper was published in which a laboratory had used this kind of germline editing in monkeys. These are animals that are fairly close to us evolutionarily. And these animals had received the CRISPR Cas9 gene editing molecules in monkey embryos, in which a particular gene was altered and none of the other genes in the animal were affected. And when those DNA-edited embryos were implanted into a female monkey, she gave birth to monkeys that were perfectly normal, except that they had this little change to their DNA. And so for me, when I saw that publication, I felt 2 reactions. I thought it was incredibly interesting scientifically, but also a bit scary, because it seemed likely to me that if one could do this in a monkey embryo, why not in a human embryo? And in fact, I started to wonder, I wonder if somebody somewhere in the world is already thinking about doing that, or even perhaps proceeding to do it. And so this really motivated me to begin a public conversation about the ethical use of germline editing, especially for this kind of application in humans, and led to an early meeting in 2015 that motivated meetings that happened since then and led to this recent National Academy report that was released. And I think is starting a very important public discussion about a technology that will truly be impactful in our worlds and gives us this awesome potential to affect the future of the human race. And this was actually this is an image taken from a recent cover of The Economist magazine under the banner Editing Humanity, in which people are already starting to speculate about using this technology not only for curing genetic disease, which I think many of us would see as a worthy goal, although it must be approached cautiously at the germline level, but going beyond that, thinking about, could this technology someday be used for creating traits that we might consider to be enhancements? And so this is really has, again, I think, very profound ethical implications that are causing many people to start to really pay attention to this technology and think about how to engage in a public conversation about how we ensure responsible use in the future. So that's really what I wanted to tell you today scientifically, and I want to share just a couple of other thoughts with you about how we move forward with a technology that is both very, very exciting, but also has profound implications. So first of all, I want to point out that the development of the CRISPR technology was and is a team effort. So it started in academic laboratories with professors and their students. We're now seeing clinicians adopting this, other researchers at institutions elsewhere. We've got a number of governments engaging and regulatory agencies getting involved, taxpayers whose money has supported some of the research that underlies this technology, and increasingly companies, commercial operations that are getting going to deploy the technology and develop it, especially for things like human therapeutics, something that's very hard to do in an academic setting and where we really need commercial operations to do this. And I myself am engaged in several companies that are doing this, as are a number of other scientists that have been involved in the early stage development of the CRISPR technology. I also want to point out that, in my own world, there are a number of affiliations that have contributed and will continue contributing to the development of the CRISPR technology going forward. And as you can see here, these include academic organizations, nonprofit organizations, the National Institutes of Health, which is funded by taxpayers in the United States, the National Science Foundation, same thing, and then a number of companies that are harnessing this technology for clinical use and for uses in agriculture and elsewhere. And finally, I just want to point out that one of my real interests has been to find a way to continue to develop the technology and the research around it for uses in various kinds of organisms, but also to really engage with the public and to play what I think is an essential role for education and outreach. And this really involves a group called the Innovative Genomics Institute, a partnership between the University of California at San Francisco and the University of California at Berkeley. I think I have to click here, you see that. And so it's a partnership between these two terrific campuses located across the bay from each other. And I invite you to get involved, visit us on the web, and learn more about the ways that we are partnering with different groups to educate the public about this technology and its uses, and to ensure responsible use going forward. Thank you very much. So I think we're now opening it up to questions, and I have some questions popping up on the screen here. And so I'm going to read out these questions. They've been sorted according to their frequency, I guess, of which they're coming in. So the top question here is, when will ordinary people be able to edit their genes to gain super athlete capabilities, e.g. Sherpa hemoglobin? Boy, that sounds great. So, I think we're still quite a ways off from that. One of the reasons that we can't do that right now is that, for the most part, we don't know what genes we would need to alter to create the Sherpa capabilities. So I think we're inhibited largely, if not for any other reason, by our lack of understanding of the information encoded in the human genome. But of course, that will change over time, and this is why I've been encouraging a very active discussion around these sorts of ethics, so that we're ready when that time does arrive. Okay, the next question is, what scares you the most about CRISPR? And I guess I have to say that, what we've seen over time, and if you follow sort of the development of new technologies, this is really true across different fields, but certainly in biotechnology, you know, sometimes we see people getting out ahead of the technology. And in other words, getting so excited that they start to deploy it before it's really ready for a particular application. And I guess, you know, there's a number of things that I find a little bit scary about CRISPR, but the thing that I worry about the most is that kind of overextension, and that might lead to some kind of a harmful effect that would then turn the public against what I think is a very, very important and promising technology. So I think many of us in the scientific community are working to ensure that that kind of getting out ahead of ourselves doesn't occur. Okay, another question is, are you concerned about CRISPR being used in biological weapons? So this question comes up not infrequently, and you know, in fact, there was a time when my inbox was full of notes saying, "Did you see that the United States has declared CRISPR to be a weapon of mass destruction?" And that's really true. It really did happen. And the thing about this is that, sure, it's a powerful technology and could be deployed in ways that could lead to creating, super bugs in principle. But we have a lot of other technologies that are equally dangerous, I would say. So I don't really worry about CRISPR for this kind of use any more than I would worry about lots of other technologies that are available, frankly. What are the main barriers to commercializing CRISPR? And there, I think, it's fascinating to me that, the technology has moved forward so quickly. So, of course, companies will have to get rights to use the technology, so there's an ongoing patent dispute that will have to be sorted out eventually. But the good news scientifically is that because it's a technology that's so widely available, any company or researcher can get access to it for research purposes, we're seeing very quick and rapid advances made both in the capabilities of the technology and the kinds of applications that it's deployed for. So I think that, currently I see that, that's something scientifically that's occurring very fast. And the barriers are ones that are just sort of common to all of us that are using this, including how we deliver the CRISPR molecules into cells and tissues, and how we control the way that DNA is repaired after it's cut by the Cas9 protein. Okay, another question is, what implication on transplant medicine do you anticipate, given the rapidly expanding hopes for the use of CRISPR Cas9? So I think that there are both companies and academic groups that have been forging ahead with one of the ideas that I mentioned when I talked about applications of this, which involves being able to make edited animals, gene-edited animals, that will be better organ donors. And this is being done in particular in pigs right now. And so I think this is really very exciting, because you may know that a lot of people that need organ transplants wait for a long time on donor lists, or maybe never are able to get access to a donor in a meaningful time frame. And if we really had a system for generating organ donor animals that were creating organs that would not be rejected by a human patient, I think this would be very, very exciting indeed. And right now, that's certainly forging ahead quickly on both fronts. Will this be the first step to immortality? Boy, I wish. I think that, again, I would just point out that, if we knew the genes to change, maybe we could do it. But right now, who knows? When will the power-up sliders for the Westworld robots be possible? Not for robots, but for us. Unless, of course, we are the robots we've been waiting for. Your guess is as good as mine on that one. We'll talk later. Okay, and then this is a good one. Are you working with ethics scientists and philosophers in developing your research? And the answer is yes. And I think this is really, really important. So one of the things that this whole experience has taught me is that it's essential for scientists to be engaging with people outside of our immediate sphere. And that includes ethicists, philosophers, and many others who have perspectives different from our own, who want and need to engage with scientists. They want to understand what it is we're doing in our labs, and they want to think about how our discoveries and inventions can be deployed in ways that will have real impact in human society. And I found this to be an absolutely fascinating aspect of all of this that sort of surprised me in a way. I found it really exciting, and I've met incredible people that are very dedicated to ensuring the rapid but also the responsible use of technology. Yes, okay, this is a good one. Do you think gene editing will create bias among humans as someone can be created to be more perfect versus someone born as is, just like the movie Gattaca? And, and again, I think it really does come back to first of all, scientifically, just the issue that we don't have the capability to do anything of that nature right now, and it'll be a while before we do. But nonetheless, knowing that we have a technology that in principle allows those kinds of changes to be made to human DNA in heritable ways, I think does, make us really think deeply about what it means to be human. What do we value about human society? And I know for myself, one of the things I find most wonderful about human societies is its diversity. I think that's precious, and I think we would never want to do anything to destroy that. So I think, in the future, going forward, and we're talking here more like decades rather than days or weeks or months, we'll have to continue this conversation. We'll have to continue thinking about what it means to be human, what we value as human beings, and what kinds of changes to our DNA we would consider to be acceptable versus those that we might not. How do you envision our civilization will use genetic manipulation and rewriting in 50 to 100 years? Wow, yeah, I think about this a bit, and it's hard to answer that question because, frankly, the science and the technology is moving so incredibly quickly. And, you can imagine that in 50 or 100 years there will be, the descendants maybe many times over of this CRISPR technology. But nonetheless, I think that we're at a very interesting point in time where we can see that this capability is going to profoundly change the way that we interact with our environment, organisms that we use for farming and agriculture and in many other ways. And so I think that over time we're going to see, what I hope we'll see is responsible uses of gene editing that allow us to do things like deal with global hunger, address challenges of climate change, address challenges of the spread of disease, things that I think will have a very positive impact on many people around the world. That's certainly what I hope for. Okay, here's a good one. Our technology is growing exponentially, but we as humans can only think linearly. How can we prepare for the power of the technology that we create? So this reminds me of several recent conversations that I've had, and including here at this conference, about the fact that human societies are very complex, and decisions that we make involve integration of data and information from many different sources and different types of expertise. And so how do we tackle that? And I don't have an easy answer for you. I think having meetings like this are a very important way to bring people together from different backgrounds to tackle and think about future challenges. But it's going to involve an ongoing set of conversations. And I think as an academic, I'd like to see changes in the way that we educate students so that they're better equipped, really, to address complex challenges in decision making in the future. Okay, so here's one. Can you edit the DNA of someone who is already mature, or must it be done while they are still in a developmental stage, like an embryo? And the answer is, so this is a really important question, because I do want everyone here to understand that you can absolutely conduct editing in someone who is already mature, or any kind of organism for that matter, in ways that don't lead to heritable changes in the DNA. And in fact, for the clinical applications that are currently being considered and developed, all of them involve that kind of gene editing, not embryo editing. So it's absolutely possible to do this in mature organisms. What kind of ecological consequences will there be from essentially controlling evolution by changing the traits of organisms by choice? So here I would like to point out that humans have been, basically have been doing gene editing kind of without knowing it for a long time in the form of breeding and selection. So we see this in particular with dogs or with plants. For really millennia, it's been possible to select for organisms that have desired traits by simply breeding them and then taking the animals that have those desired traits and allowing them to continue to breed. So in that sense, we've clearly been controlling the evolution of organisms that we interact with that way by simply selective breeding. So I think what the CRISPR technology does is it just gives us a way to speed up that process, make it much more precise, and gives us tools with which we can understand the information encoded in DNA much more quickly, and thereby know which kinds of mutations will be beneficial to make. Yeah, how can we make sure that this will be available to rich and poor alike? Very, very interesting question and very important. And I think, when you think about clinical uses, it's hard to imagine, at least today, how that could be made equitable to folks around the world, just because of the cost of any kind of sort of personalized type of technology like this that might be used for particular individuals. And I think that here I'd like to just point out that one of the incredibly interesting things about the CRISPR technology is that in addition to being used directly as a clinical treatment for disease, we can also think about deploying it in ways that will prevent many people from ever being exposed to a disease. And I gave the example during the talk of something called gene drive, a way of using the CRISPR system to drive a trait very quickly through a population of organisms. And this is already being tested in insects, and we can imagine doing it in ticks, for example, that cause Lyme disease. So I think that, for me, when I think about that question, I really think about ways that we can envision using gene editing to protect people from disease across broad populations, and maybe to do things in agriculture that will enable more productive and nutritious plants to be grown, even in the face of things like climate change. So that's it, folks. Thanks very much for attending, and see you at the conference.